Okay, so the last trichrome stain that we're going to troubleshoot is the MSB stain. If we begin with this slide here at the moment, this actually shows the outcome that we expect to see. So we have good, strong, defined nuclear features. There's quite a variety of cell morphologies in this section of placenta. But in any case, those nuclei are really nicely demonstrated. The fibrin being demonstrated by this red staining, the scarlet red, is nicely demonstrated. If we scan around, we can see other areas of fibrin there as well. And the collagen has stained up really nicely as well. The other thing that we can notice is with respect to the red blood cells, the majority of the red blood cells clearly stained a yellow colour, and that indicates that the slide has been post-fixed in the picric acid for the best part of an hour, at least 45 minutes to an hour, to achieve that nice even density. And so, yeah, this is a really nice slide. I think I've given that about a 7. And so that's what you would be aiming for when performing this stain. Now, I mentioned the advantages there of post-fixation. Let's have a look at another slide. It's actually from a different block, but it is one that is noted here to be not post-fixed. And so hopefully we can see some evidence of that with respect to the quality of staining. So the first thing that we can see is that in the absence of post-fixation, or perhaps it only had limited post-fixation, there is now a mixture of colours for the red blood cells. So just in this area here alone, we can see there's red, there's blue, as well as the yellow, and that's simply due to inadequate fixation of those red blood cells, resulting in uneven cross-linking. So there are some cells have larger gaps within it, others have more closer cross-links due to being more fully fixed. And that's what you end up with, this variety of colours due to differential partitioning of those various dyes within those spaces. The other thing that we can notice here is that the collagen is generally pretty good. However, with respect to fibrin, we often find, as for the red blood cells, that you get a more even density when it has been post-fixed. So on this side here, there is some fibrin which is quite pale and certainly has less of the scarlet red. Now, bearing in mind that fibrin does present in varying levels of density, which reflect, I guess, the age and varying amounts of fibrinolysis that take place. So it's okay to have a little bit of variable staining, but often you will find that there can be less staining of fibrin, or less red staining in general, without the post-fixation technique having been employed. So that's certainly something to watch for. And even when the slides have been well post-fixed, you need to be careful with your PTA. So PTA is obviously applied with the goal to clearing the collagen of the scarlet red in preparation for the methyl blue. But if you overdo it, then you can result in loss of some of the fibrin staining, and that's not too good. So as way of example of that, this next slide has a number of problems going on with it, including most likely over-treatment with the PTA. So just to bring that into focus, that's approximately in focus there now. So the first thing I can see here is that there's reasonably good collagen staining, as evidenced by that nice blue colour there. There is very consistent, almost all of the red blood cells are stained yellow. There's 1 or 2 blue red blood cells, but the majority have been stained quite well. So the fact that there is a very even staining of those red blood cells indicates to me that this has been well post-fixed, most likely for the best part of an hour. That is probably also enhancing the level of collagen staining. The two things to me, however, that stand out as not being good is the fibrin, which I can hardly see, and the cell nuclei are also very pale. So let's try and find an area where there should be fibrin, and then just consider whether or not it even got the scarlet red. Maybe it was stained upside down. Okay, so there's an area here which seems to be similar in morphology to the areas of fibrin in the other slide, and I can actually see there's definitely traces of red. Just move that up on the screen so it's a bit clearer. Okay, so there's definitely a hint of red being present within those areas that morphologically resemble the fibrin clots in the earlier slide. If we look even more closely, some of those areas actually look yellow rather than looking red. So most likely what's happened here is that the brief rinse in water after the scarlet red has maybe been a bit too excessive, and that could have removed some of the red, but probably it's the PTA that's been left on for too long. So perhaps the student on this occasion has been unaware of the level of staining that's appropriate, and upon seeing the red there, they probably thought they need to keep on clearing it until most of it has been removed. But unfortunately on this occasion that's resulted in the fibrin becoming pretty much colourless. And given that that's the purpose of the stain, that's unfortunate. So, you know, students often ask us to grade or rate the slides as they're being presented. You know, this would clearly be a slide that would receive a failing grade, simply due to the fact that the purpose of the MSB stain is to demonstrate fibrin primarily. Yes, we like to see the other structures as well, but our inability to see the fibrin clearly to any significant level would result in a grade of probably 3 for this particular slide. Just finally, looking at the nucleus staining, there is a degree of nucleus staining that can be seen. It seems to be the right colour in terms of being a kind of a sort of black, almost, well, grey in this case. But because the nucleus staining is so pale, the most likely explanation for that is that the nuclei were over-differentiated to begin with. So following the 10 minutes in the Weigert's hematoxylin, it's no doubt had maybe 3 or 4 dips in the acid alcohol as opposed to the mandatory 1 dip and 1 dip only, and that's just reduced the overall staining with the hematoxylin. So apart from that, we can see the collagen quite well, the red blood cells are staining quite well, but in the absence of seeing fibrin, that's really not too good. Okay, so look, you know, this is a very limited demonstration of things that can go wrong, but hopefully it just starts you thinking about some of the problems that can occur when performing trichromes. Take-home message, make sure you get your nucleus staining right, 10 minutes in the Weigert's hematoxylin, 1 dip and 1 dip only in the acid alcohol, blew it, and then you're off to a good start. From there, it's really about paying attention to the solubility of these various acidic dyes that are being applied. So with the Van Giesen, you know, make sure that you're blotting after staining, make sure that you then go straight to the absolute alcohol and keeping those rinses brief. And when it comes to these more complicated stains, you really have to use the microscope control after each step to ensure that you've got the right level of staining prior to moving on.